Hello everyone, and welcome to The Explainer. Today we're going to break down a topic that's right at the heart of all modern medicine. Let's get started. It's the million-dollar question, right? We see headlines about medical breakthroughs every day, but how do we get from a promising idea in a lab to a treatment that you and your doctor can actually trust? How do we separate real hope from, just hype? Well, the answer lies in this key term, pivotal evidence. Think of it as the gold standard, the undeniable proof. It's not an opinion, and it's not a gut feeling. It is the solid, robust evidence that regulatory agencies require to prove that a new drug not only works, but is safe for the public. So to understand how this evidence gets built, here's our roadmap. We'll start with the challenge of gathering evidence, look at why a control group is so critical, and how we design trials to be bias-proof. Then we'll get into the nitty-gritty of defining the question with something called the estimand, see how it all works in practice, and wrap up with how this all builds scientific trust. All right, let's start at the very beginning. The first big challenge is, how do you even measure the effect of something if you have nothing to compare it to? And that's the absolute crux of it. To know if a new treatment is working, you have to compare it against something else. This is where the control group comes in. One group of patients gets the new medicine, while the control group gets something else. This comparison is the foundation of any rigorous clinical trial. Without it, all you have are stories. And there isn't just one type of control. Sometimes a placebo, a substance with no active ingredient, is used especially if no treatment exists. Other times, and this is really important, the new drug is compared against the best treatment that's already available. That's called an active control. The choice really depends on the disease, ethics, and the exact question you're trying to answer. Okay, so we've got our groups to compare, but how do we make sure that comparison is truly fair? How do we keep our own expectations or any other factors from messing up the results? The great enemy of science is bias. Bias is anything that can unfairly influence the results, making a treatment look better or worse than it really is. Luckily, science has two incredibly powerful tools to fight back.
First up is randomization. It's basically like flipping a coin to decide which patient goes into which group. This ensures that, on average, the groups are as similar as possible in every single way, except for the medicine they're receiving. The second tool is blinding, where neither the patient nor the doctor knows who's taking the real drug and who's in the control group. It's a brilliant way to remove our own hopes and expectations from the equation so we can focus only on the drug's true effect.
Now, let's go a little deeper, because the real world is messy. Not every patient follows a treatment plan perfectly, so what do we do then? Modern trial design has evolved to face this complexity head-on.
Exactly. Imagine a patient in a trial stops taking the drug because of side effects, or their condition gets worse and they need an extra rescue medication. These things, called intercurrent events, can really muddy the waters and make it tough to figure out the drug's real impact.
The solution to this is a newer but absolutely critical concept called the estimand. It was introduced to force researchers to be incredibly precise. An estimand defines exactly what effect you want to measure, in which population, and crucially, how you're going to handle those messy real-world events right from the start.
The cool thing about the estimand is that it forces you to nail down four key things: the population you're studying, the variable you're measuring, how you'll handle those tricky intercurrent events, and finally, how you'll summarize the overall effect. It's all about getting rid of any ambiguity so that when you read the trial results, everyone knows exactly what question was answered. So, having a perfect plan on paper is one thing, but executing it flawlessly in the real world with real patients and real doctors is something else entirely. For that, you need rules. And the golden rule, the guiding principle for everything, comes from the Good Clinical Practice Guidelines, or GCP. This is the ethical bedrock of all clinical research. It reminds us that above all else, the safety and well-being of trial volunteers are the top priority. The entire scientific machine serves this principle.
Good clinical practice is essentially the trial's instruction manual. It defines everyone's responsibilities, from the investigator treating the patients to the sponsor funding the study. It covers everything, following the plan to the letter, reporting any side effects, and making sure the data collected is of the highest quality. It's the guarantee that the trial is run ethically and rigorously.
And so, by putting all of these pieces together, we arrive at the verdict. A verdict we can trust because it isn't based on chance, but on a clear, scientific method.
So this is the complete journey. It starts with a clear clinical question. You create a rigorous experimental design with a control group and randomization to prevent bias. You define a precise estimand to handle real-world complexities. You run the trial, following the strict rules of GCP to protect patients and ensure data integrity. And only then, after all that, do you analyze the results and reach a conclusion.
And here's the fundamental takeaway. The pivotal evidence that supports a new drug's approval isn't just a nice story or an expert's opinion. It's the final product of this methodical, transparent, and scientifically sound process. It's a system designed to give all of us, patients and doctors alike, confidence that a medicine's benefits truly outweigh its risks.
And to leave you with a final thought, here's a question being debated in scientific circles right now. With the rise of real-world data, information collected from electronic health records and wearable devices, could this model change? Could this new data complement, or maybe even one day replace, parts of this rigorous process? It's a fascinating field that's constantly evolving. Thanks for joining us on The Explainer. Stay curious.